Okay, I want to go over your lab real quick. So you should have, or you will be getting, a nice big old thick document here. Basically, you have some serums and you have some simulated blood. Of course, none of this blood is real. There's different ways you could actually make this lab from household products too, with just various acids and bases. But regardless, ours is from Ward Scientific, so none of this is toxic. You have a sheet of tons of vocab words. You have a little information on the background of ABO blood typing, and if you've done tutorial 1 and 2, kind of goes right along with that. So seniors, you will need to make sure you go back and do tutorials 1 and 2. My sub will give you that document. Okay, so then you get into, because there's so many A words in ABO blood typing. There's antigens, there's surface proteins on the red blood cells, then there's antibodies that are floating around in plasma. Then there's another A word called agglutination, and so that is clumping. And this figure 2 is kind of like your key. So as you run your test, if you see agglutination in one of the wells, but not in the others, then that's going to tell you that it was positive for that. So that would be like type A, and I'm going to kind of explain that to you. Okay, so that is what you're going to refer back to. All right, so then you have your pre-lab questions. You've got a little scenario. Okay, we are not doing the microscope. It's not really, no. So then you'll get to part B, ABO blood typing, and so you are going to have these are the trays. They look like this, and they are labeled, and it says A, B, and RH. I think it helps because these tables are so, like, they've just got too much color texture. I think it helps to put them on a white paper or paper towels, and then you can also then just mark on your paper. Okay, so it helps because then you can just label them right here on the bottom. So all right, so you're going to have a tray for each different blood. So you're going to have crime scene, victim, suspect 1, suspect 2, suspect 3, suspect 4. So you're going to have 6 trays, and you would definitely want to label them. So then you're going to come over here and you're going to grab the blood, and the blood is labeled as right there, suspect 3, suspect 1. All right, so again, you have to make sure you keep track of all this, and you're going to put about 3 drops of suspect. You know, you're going to do all these, but right now I've got suspect 3 in my hand. So I'm going to put 3 drops in each of these wells. Okay, so you will do that for all 6 of your blood types, or the different types of blood. 3 drops in each of those. Then you have the different antibody serums. So which one says A? All right, so this says anti-A serum. So that is only going to go in the A well. All right, so I'm going to put 3 drops. So my blood is already in here. I'm going to put 3 drops of anti-A on. Then I'm going to put 3 drops of RH serum in the RH well. And then I've got B that's going to go in B. And then I've got these little disposable, we're actually going to wash these off, toothpicks. 1 per, don't cross-contaminate. You'll stir it up. And when you stir it up, if it stays clear and red, then there was no reaction. If it agglutinates, it's going to have kind of a cloudy or maybe dotty appearance. And if that's the case, so if it agglutinates in A, but not in these 2, then it's type A blood. Okay. So again, you would go back to your chart right here. Okay. I'm trying to think. So then you have your observation sheet, and then you have your data chart down here at the bottom. Okay, so you know, agglutinates, yes, no, and then you will say blood type. So you'll be A, you'll be B, A, B, O, and then the RH would be plus or minus. Okay, so if it doesn't agglutinate, then it would be RH negative. So if the only thing that agglutinates here is A, then it would be A negative blood. Okay, if it agglutinates here, then it would be A positive. Alrighty. So when you're finished with all of those, then you will wash these up, and then you will wash off your red toothpicks and turn in your lab. Okay, when you finish that, then we're going to do tutorial number 3. Okay, all right, I will not see you the rest of this week, so have a good weekend.